Welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter Microbes in Human Welfare. Part 2, Microbes in Industrial Products. Microbes in Industrial Products. Even in industry, microbes are used to synthesize the number of products the valuable the human beings. So here the number of the microbes are using as the number of ways in industrial. They are to development of what, like as in beverages, antibiotics. These are the examples. So the production of the beverages as well as antibiotics we are using as in microbes. Here the production on industrial scales and requires growing microbes is very large the vessels called as any fermenters. So in the industrial process, we are developed as the large number of their products. Here vessels that completely we called as what, that is in a completely fermenters, fermented beverages. Microbes, especially yeast, has have been used from the time of immemorial for as in production of beverages like wine, beer, whiskey, brandy, or rum. So here yeast mostly we are using as in beverage industry. The production of what, wines, beer, whiskey, brandy, rum, these all things. And the purpose of that some yeast we are using the Saccharomyces cerevisiae used for as in the bread making in that in a commonly called as in a beverage yeast. They use it for as a fermenting their melted cereals and fruity juices to the production of ethanols. So here yeast bacteria Saccharomyces cerevisiae we are using as in a baker's yeast, and then here some microbes are used as in a they are melted cereals and fruity juices as production of ethanol's purpose also we are using as in some microbes. Okay, yes. Do you collect the metabolic reactions which result in the production of the ethanol by yeast? The process of when we see that as in a glucose they participate in the fermentation process, they produce as a ethanol as well as in a carbon dioxide is releasing, correct? Yeah. Next, depending upon the type of the raw materials used as a fermentation is a type of processing, they are with or without distillation. So when your production of the what they're completely different types of the alcohols we are the raw materials used we developed as an alcohols, right? There are two types, with and without a distillation. This type of alcohols drink as obtained. So the different types of alcohols drinks we are obtained with the help of what, incomplete any microbes. Okay, this is next. The wine and beers are produced without a distillation. They are, whereas whiskey, brandy, rum is produced by distillation of in the fermented broth. So here completely with the distillation, without a distillation. The photograph the fermentation plant show as a figure 8.5. So 8.5 they are completely given as any fermented entire diagram. Antibiotics. Antibiotics produced by microbes are regarded one of the most significant discoveries of the 20th century, have greatly contributed towards the welfare of human society. Here, their antibiotics. The antibiotics produced as a large is most significant in the 20th century that human society. Here, anti means that is a Greek word that means against. Bio means life. Together, we call as an against life. In context of in a disease causing organisms. So the disease causing organisms they get fight to get against, that is we call as an antibiotics. Yeah, whereas with reference to the human beings, they are pro life and not against. They are pro life, not against. Antibiotics are chemical substances which as in they produced by in some microbes and can kill or they are completely retard the growth of other like disease causing microbes. So some microbes are get helper as to get controlled or to get completely kill that in a unwanted diseases. Okay, yeah. Like next, You are familiar with as in a common disease antibiotics, that is penicillium. Do you know that penicillium was first antibiotic to discovered by the Alexander Fleming? The chance of discovery. The Alexander Fleming, while the working the in a streptop, here very important, this staphylococci bacteria. Staphylococci bacteria, very important. The Alexander Fleming, he first discovered as in that penicillium, the bacterium is staphylococci bacteria. Once observed the mold growing one of his unwashed culture plates around which as in staphylococci could be not grow. So he took as in the two culture plates, one is unwashed, one is in a washed. So here completely there. Once he observed as in a growing one of his unwashed culture there, which is staphylococci is not grow. So unwashed did not grow. Here he noticed. Next, he found out that was due to that chemical production the produced by as in a the mold and named as in a penicillium after mold penicillium notatum. So after he noticed as in they due to the formation of the penicillium, after he called as in a mold we called as penicillium notatum. That is the name is what? Penicillium notatum. Let's see. However, the full potential and as the effective antibiotic was established much later by the scientist Ernest Chain and Harvard Florey. So the number of here in this antibiotics, the potentially effectively antibiotic established the scientist was Ernest Chain and Harvard Florey. Both scientists they did as a series of experiments on the antibiotics. Next, the antibiotics was extensively used as a treatment of American soldiers and wounded in World War II. So in the World War time, the number World War II at the time, the number of soldiers there causes a number of wounded. So there in this antibiotics, the first time they get used. Okay, the Fleming, Chain, and Florey were awarded Nobel Prize in 1945. In 1945, he get as any their Nobel Prize for this discovery. For this discovery, this is next. After this penicillium, other antibiotics also they're purified from other microbes. Here after penicillium, other microbes we are also purified from other microbes. Can you name some antibiotics? Find that is in a source. So when you see that as in the number of the other antibiotics, ampicillin, tetracycline, like here, the antibiotics have the great improve our capacity to treat their delay the diseases such as in a plague and whooping cough, and nothing but as a colignancy, diphtheria, and leprosy, like nothing but as a Kushtu rog, which is used as a kill millions all over the globe. Today we can't imagine without antibiotics the world. So this total number of diseases they get controlled by as in these antibiotics. Nowadays we can't imagine the world without antibiotics. Okay, these are next chemicals, enzymes, and other bioactive molecules. Microbes are used as in a commercial and industrial production of the certain chemical organisms like as an organic acid, alcohols, enzymes. So here some chemicals, enzymes, alcohols were used to the production where we are using as in microbes. Examples here, the acid producers like Aspergillus niger, it is in a fungus of the citric acid. And Acidobacter aceti, the bacterium, it is of acetic bacteria acid. Acetic acid get produced by as in the Acidobacter. And next, Clostridium bacterium, it is a bacterium of acidic acid, and lactic acid, lactobacillus bacterium is lactic acid. So different bacteria to get producing as different types of acids. Okay, here most of the yeast Saccharomyces cerevisiae used for as a commercial production of ethanol. Yeast used for as in to get production of what? In ethanol. And microbes, see ethanol. And then the microbes are used in the production of enzymes, that is a life, that is the microbes are also used as in a production of the enzymes. Lipase are used as a completely the detergent formulation of harmful remove oil stains from the laundry. Here lipases get using as in a to removal of oil and the laundry oil stains and from the inner laundry. Next, you must have the noticed the bottle juices brought that in market in a clear as in a compared to as other made home. So when nearby the roads, their bottle juices very clear because here this because their bottle juices are they are clarified with using as a pectinase and protease enzymes. So in that bottle juices, in that here the bottle juices they get added with as what? Protease and pectinase enzymes. Protease as well as pectinase enzymes to get used. Next, streptokinase, the production of the bacterium, the streptococcus, it is modified. The genetic engineering is used as a their clot blaster to removal of clots from blood vessels. Patient who are under myocardial infection leading to as in heart attacks. Here, streptococcus, the streptococcus that enter we get used as in a the clots to get clot bluster means the myocardial heart arrest that when they make as any the clots to get easy to get used as in the streptococcus to remove as in that enter the clots. Okay, next another bioactive molecule that is cyclosporin A. The cyclosporin A that is get used as in that in a immunosuppressive agent. It is an immunosuppressive agent organ transplantation of patients. Immunosuppressive agent that is cyclosporin A. It is produced from fungus that is a Trichoderma polysporum. That used as in a Trichoderma polysporum. We are obtained from this cyclosporin A. Now, here stannins, the produced by the in yeast Monascus purpureus has been used as a commercially the blood cholesterol lower agents. So here mostly the Monascus purpureus we are using as a commercialized to blood cholesterol lowering agents. It acts as in a compactively inhibiting enzymes responsible for synthesizing cholesterol. So cholesterol to get control purpose we are using as in this one. Okay, these are the enzymes and bioactive enzymes. Okay, now remaining to discuss in next class. Thank you for watching. Please like and share. Thank you.